The first speaker we have is Dr. Asa Bradman. Asa is a longtime buddy of all of us. He actually used to be working for the state years back. He's an environmental health scientist at UC Berkeley and worked with Dr. Brenda Eskenazi to co-found the Center for Environmental Research on Children's Health in 1997. And he has since done lots of work, great work. He currently is helping direct the biomonitoring and exposure aspects of the CHAMACOS study in the Salinas Valley in California. He's co-principal investigator of the National Children's Study in Kern County, and also leads an initiative to improve environmental health in California child care facilities. So Dr. Bradman is going to talk to us today about current issues in childhood exposures to environmental toxicants, case studies in agricultural communities, aka the Salinas Valley, and California child care centers. Thank you, Asa.
So I'm just going to start making a few comments. One, I decided to focus this presentation a lot on challenges in exposure assessment and kind of implications for risk assessment and epidemiology. So I'm not going to talk a lot about our health effects work. I'm not going to talk a lot about stuff we've done on peroxidase or genetic polymorphisms or epigenetics. I'm going to focus on some biomonitoring issues. And I think, though, they're very relevant to OEHA and risk assessment. If I just heard what I heard a moment ago from George, I probably would have put more emphasis on the child care work we're doing, but I will highlight a few things here. So again, I'm Asa Bradman from the Center for Environmental Research in Children's Health at Berkeley. So again, the talk today is going to focus on pesticide exposure issues that we've kind of uncovered and have been addressing as part of our CHAMACOS cohort studies, and then challenges and what implications they may have for risk assessment and epidemiology. Just a reminder, I don't think we need why children are more vulnerable to environmental exposures here, but just quickly, people get exposed, ingestion, inhalation, dermal, and then we shouldn't also forget the prenatal exposures across the placenta, and those are very common. And as we know, children are more exposed than adults in a given environment, and they're also more vulnerable. So to shift to CHAMACOS now, CHAMACOS is a community-university partnership. We started in 1997-98, and we have a number of objectives, but primarily the focus really is the environment in general and its effect on children's health and development. So we're looking at pesticides, we're looking at flame retardants, we're looking at BPA, a number of different compounds, we have a number of different outcomes. We're looking at neurodevelopment, growth. We also have some asthma work we're doing that we haven't really developed that as much as we could have yet, but hopefully that will move forward. And now we're looking at a lot of outcomes in our teenage children, pre-teenage children, obesity, metabolic syndrome, and also onset of puberty. We also have a lot of benchtop studies, and we also have been doing a lot of work on interventions to reduce exposures. So the CHAMACOS study is based in the Salinas Valley, about 100 miles south of here. This is kind of a map of pesticide use in California. And you can see the valley lights up as a relatively heavy use area. It also has a very long growing season, so the population there tends to be more stable than, say, Central Valley populations. For the core cohort study, we enrolled 600 pregnant women in 2000, primarily low-income, Spanish-speaking, Mexican immigrant population, either working in agriculture while they're pregnant or coming from families where someone works in agriculture. I want to emphasize, working in agriculture was not a criteria. This is not a farm worker study, but we're working in an area where there's many farm workers. It's a longitudinal birth cohort study. We have had lots of contacts, and they're ongoing. We're looking at a lot of health outcomes. I emphasize neurodevelopment, respiratory function. We've done a lot of home inspections. We've collected hundreds of thousands of samples at this point. We have over 100,000 samples stored. We have urine, blood, teeth, breast milk, saliva, hair, any number of different media that we're working on in different capacities. I'm going to talk a lot about organophosphate pesticides today. I think all of you here are familiar with those. They're a heavily used insecticide. They still make up a very large proportion of total agricultural insecticide use, and they're used heavily in the Salinas Valley, about 500,000 pounds a year. I want to also emphasize, and most of you are familiar, they break down into metabolites in urine, most of them, not all of them, and they come up as a dimethyl or a diethyl. These are the dialkyl phosphate metabolites. And I'm going to be talking a little bit later about differences on interpretation of those metabolites in urine. And much of what I'm talking about will also be relevant in the future, I think, to other kinds of metabolites and indicators for exposure that are looked at in urine. There's a lot of talk about pesticide use declining in California, at least OP pesticide use. And in fact, organophosphate pesticide use has declined over the years, although in the Salinas Valley, if we look from when we started in 1998, use has been relatively constant. And that's due to some specific pests that put a lot of pressure on the lettuce and other crops there. So OPs are still around, but there are changes in agricultural use, and there's also bigger changes in residential use. Chlorpyrifos and diazepam, two very common compounds, were removed from residential use. So just a quick summary now. This is over the years, a summary of the dialkyl phosphate metabolites in the urine of our mothers and kids. Just to highlight a few things here, the blue is CHAMACOS. If we look at these levels here, these are our levels during pregnancy. Don't worry too much about the numbers. And these are levels in women of childbearing age in NHANES. And you can see our levels are about 40% higher. They're not 10 times higher, they're not twice as high. So they're in the same range as the general US population, but there is some suggestion here of higher exposure. If we look at the trend with kids, the levels go up as they get older. When we compare them to the youngest age group measured by NHANES, 6 to 11, once we get up to 3.5 or 5 years, we're the same or even a bit higher than the nationally representative samples. So there's some evidence here of higher exposure in our population, and this is consistent with some work done by Martha Harnley, where she looked at nearby pesticide use and pesticides in household dust, and particularly for persistent compounds, there were relationships between nearby use and higher levels in the dust. Chlorpyrifos, iprodione, dactyl, herbicide are all very persistent, and I think in those cases particularly you get a stronger signal between the source and the measurements in the home. We've also done some modeling trying to look at those dust levels, look at the PUR data as a source of pesticides, then making some assumptions about transport into the home, transport out of the home, and basically putting together a fugacity model. This is work done by Tom McCone, and estimate what would be the likely result in the receptor, in this case, the mothers. And when we compare our modeled estimates based on environmental measures, primarily dermal, and hand-to-mouth, of course, is very low. These are for the pregnant mothers. And then other non-dietary ingestion or inhalation, and then estimated food exposures, we actually, when you add these up, we come pretty close to our actual measured metabolized dose. So this was an effort to really take a modeling approach here and then compare it to an actual measurements and see if we're in range. And we definitely are in range, and we're certainly, actually it's remarkably similar. And we're now trying to transfer this approach to the children as well and see if we can also reproduce with kind of validate the modeling with the measurements. But again, there's kind of a suggestion here that the environment is important, but food really comes up as a primary source of exposure, in this case to the mothers, and that started to get us thinking about how important food may be relative to other sources. And we spent a lot of time now looking at that. In our children, these are looking at our younger kids now, 6 to 24 months of age, consistently, those who ate more fruits and vegetables had higher metabolites in urine. And they're statistically significant up here, but there's a very consistent trend where we have higher metabolites in kids who eat more fruits and vegetables. Kind of our next step when we were puzzling over this was to do basically a study like Fensky and others have done in Seattle, was to essentially do a clinical trial of sorts where we had them a group of children eat organic food and see if there were changes in levels. So there's a couple of things going on here. We did this in Oakland and Salinas. The idea was here to compare both a rural and an urban area, and then also look at the effect of diet. So a few things here. If we look at the conventional period, the levels in Oakland were consistently lower than the levels in Salinas. So when they were just having a regular diet before we introduced the intervention, the levels were higher in Salinas, which is consistent with our thought that there's a local contribution. although again, if we also look during the organic week, the levels did go down, but they were still higher in Salinas relative to Oakland. So still we have some evidence here. I mean, we have evidence here that there is a local contribution from agricultural use, or likely from agricultural use, but there's also a big contribution of diet here. And in fact, the contribution from diet in Salinas was higher than Oakland. So there's kind of some different things going on here. I think this may have to do with taking produce from the fields, relatively higher levels. And when we gave them organic food, they probably had much lower levels, probably even relative to Oakland, since the people in Oakland are probably buying food that's farther in time away from harvest. But again, the implication here is that diet is important for exposure, and location is also important well. We have another study, and I'll talk more about this later, where we have a small group of kids where we took samples very intensively over 7 days, and then we had a detailed inventory of exposure factors. And when we look at one variable, apple juice and actually other fruit juice, we have a remarkable association between how many servings of apple juice they've had in the previous days and the levels of metabolites in their urine. In fact, the R-squared here is around, in this particular analysis, is around 0.5 or 0.6, and it's pretty amazing to have that proportion of a highly variable outcome explained by a relatively simple variable. So diet seems to be very important in explaining these metabolite levels in urine. Shift a little bit over now, looking at occupational exposures. This is a study we did with strawberry harvesters. And I'm just going to focus on the blue line here. This is I kind of like these graphs. It's kind of a cumulative frequency distribution graph. If, in this case, the metabolite levels are on the x-axis, not the y-axis we're more familiar. So if you're shifted to the right, you have higher levels than the population to the left. So if you look at the blue, these were farm workers that were sampled in a field after working in a field that had been sprayed with Malathion a few days before. The post-harvest interval had expired and they were harvesting. And they had much relatively higher levels, particularly if we look at this yellow group here, that's levels in NHANES for this is a Malathion metabolite in urine. And they had basically no detection except maybe for the top 1%, whereas our population was much, much higher. And just to give a little explanation here, I know people have trouble with these graphs. So if we look at the midpoint here, that's the 50th percentile. So the median for the general US population is 0, but the median for our population of farm workers was about 120 at that cut point. So again, much higher exposures, in this case, occupationally related. Just very quickly, we also found lower levels of residues on their hands if they wore gloves, and that also resulted in lower levels in their urine. If they wore gloves, they had much lower levels of the metabolites. But again, we see that diet's important. These are strawberry harvesters. Some were eating strawberries in the field, and those who were eating strawberries in the field had much higher metabolite levels. So again, here's a classic example where in this population, we're looking at both occupational and dietary exposures. Then one final look at our CHAMACOS population. This is looking at the ratio of 1 naphthalene to 2 naphthalene. These both derive from a pesticide called carbaryl, a carbamate. But naphthalene itself also comes from, these compounds also may come from combustion sources, cigarette smoke, other things. I'm sorry. When these 1 and 2 naphthalene are derived from other sources like cigarette smoke I just mentioned, they come in at a ratio of about 1 to 1. You get both 1 naphthalene and 2 naphthalene. When you're exposed just to carbaryl, you get much higher levels of 1 naphthal. That's the degradant of carbaryl. So if you have an elevated ratio of 1 naphthal to 2 naphthal, that suggests carbaryl exposure, and usually the cutoff that's used is a ratio of greater than 2. If we look at women in our CHAMACOS population that worked in strawberry fields, they had a much higher odds of having a ratio greater than 2. Again, kind of underscoring that participants in our population are subject to a variety of exposures, both occupational exposures, take-home exposures. I didn't go into that in too much detail, but we have other evidence for that, and also environmental pathways. So there's kind of a plethora of stuff, of pathways of pesticides getting into our population. We also see health effects from these. And I'm not going to go into this in detail, and Brenda Eskenazi is here, is going to be talking about this later. But we just to quickly summarize, we've seen relationships between, in particular, the prenatal metabolites in the mothers and a number of outcomes: shorter gestation, developmental problems at early childhood and school age, and these results tend to be consistent with across age points in other studies. And Brenda Eskenazi, who's here, will be talking about that in more detail later. So I want to shift over to some of the challenges in interpretation that we've been dealing with and I think are important for risk assessment, particularly with respect to OEHA and other interpretation of biomonitoring data. Okay. I'm going to try to go through this in about 5 minutes. So I talked about these DAPs that we've been measuring, these dialkyl phosphates. They're class-specific, not pesticide-specific, so we don't know what the parent pesticide compound was. They were originally validated for occupational studies. They're easy to measure, and they're in samples like urine. They can be measured in samples like urine that are easy to collect. The problem of not knowing the parent compound is kind of illustrated here. This is kind of a relative risk of the different pesticides that were used in 2000 in the Salinas Valley. Oxydimeton methyl is way more toxic than the other ones. So this is the use divided by the reference dose. If we look at oxydimeton and malathion, they have almost equal pesticide use, these white bars. But if we look at the relative contribution to risk, oxydimeton methyl is off the chart compared to malathion because it's about 20 times more toxic. However, when we do a risk assessment, we don't really know what the parent compound is. We did this exercise, which you're probably familiar with, where we tried to do a cumulative risk assessment and made some assumptions about dividing up the pie of exposure based on pesticide use reporting data. And this result suggested that some of our women would be above the benchmark dose for BMD10 for OP exposure. This is putting chlorpyrifos equivalents. But the message here was that a portion of our population did exceed the BMD10, the benchmark where we'd expect 10% of the population to have, in this case, brain cholinesterase inhibition. The point here is that we had to make some guesses about what the mix of exposures were. We can't, and with the DAP, since we don't have pesticide-specific markers, we can't really do that very well. Brenda will talk about this again in more detail, but this is the relationship of DAPs and 2-year Bailey mental scores. And you'll see here that the coefficients are negative. We found an inverse association. But it was strongest for the dimethyls and weaker for the diethyls. Well, maybe that dimethyl is a marker for oxydimeton methyl, which we know is more toxic, and oxydimeton methyl devolves to a dimethyl. There could also be an issue that maybe the ethyls, which is the other half, are just bad markers. If you look at here, these are overnight diaper samples. We have spot samples and overnight diaper samples. The ethyls they basically disappear overnight. And maybe they're just chemically unstable and just bad indicators of pesticide exposure. And if you look here, this is the correlation of 24 and spot urine samples collected the same day. Pretty good for total DAPs and dimethyls. In other words, a spot sample does represent a 24-hour samples, but weaker for diethyls, again suggesting instability. These are now samples collected over days. And again, the dimethyls have lower correlations from samples collected 1 day apart, 2 days apart, et cetera. You can see, in fact, out 2 days apart, they go to 0. Although a little bit concerning here also is that when we look at total DAPs and dimethyls, a day apart, they're not very correlated, only 0.48, and then it goes down to 0 after just a few days, but a little longer than the diethyls. Again, now we look at the correlation of these DAPs in 24-hour samples collected 1 day apart and 3 days apart, and they're not very correlated. So when we collect a given measurement, we have this issue of instability in the chemical marker, but also there's just high variability. A sample collected on a given day doesn't represent exposure over a longer time period. Here's a visual of this. These are the high and low values of these 24-hour samples collected just 3 days apart. And many of these, this is on a log scale, many of these just 3 days apart are 2 orders of magnitude different. So very high variability, and a very big issue for estimating exposure, both for epidemiological studies and risk assessment. Again, back to that variability, it looks like diet is a big source. And I won't go into detail again, but again, with just a few variables, we can explain a large proportion of the variability. So I'm getting a little short on time here, so I'm going to quickly go through this. Another issue now is we've measured these metabolites, but where do they come from? The metabolites can come from parent compounds, OP, pesticides, in food or the environment, but you can get exposed to the preformed metabolite. And the urine DAPs may reflect both preformed DAPs and parent pesticides, and the preformed DAPs aren't toxic or very toxic. So when we interpret a urinary metabolite, there's issues of short-term variability and then also whether it's representative exposure. This also applies to other things we measure in urine well. We do find these preformed metabolites in the environment. In our case, we looked at dust, so they are clearly out there, and they've also been found in food. In the case of dust, we've done some modeling which suggests that dust in particular is a relatively minimal source of exposure to children, but we know from diet studies that preformed DAPs can be a significant source in food. So again, interpreting the urinary metabolites, it raises a lot of challenges, and a lot of concerns when we're doing epidemiology. So some of the implications here. One, I still need to emphasize that I think urinary metabolites are a valuable tool to assess exposure. And this was something that came up at a EPA pesticide stakeholders workshop just a few months ago. And everyone agrees that urinary metabolites are important and are going to remain important. They're easy to collect, especially for children, and there's laboratory methods available. They seem to work for adults. And again, Brenda will be going to this in more detail, but we find fairly consistent relationships, in our case, with the exposures during pregnancy and later health outcomes. But in terms of risk assessment, the high intra-individual variability that we have shown here raises a lot of issues. One, cross-sectional sampling can give a range of population exposure, but not an indicator of individual chronic exposure, which is what you need both for many risk assessment purposes and also epidemiologic purposes. I got it. And this has big issues, I think, from the National Children's Study. Then this issue of preformed metabolites, which is not specific to OP pesticides. Again, this issue of oxydimeton methyl, we couldn't measure exposure to that individual pesticide. So for many pesticides and other compounds, there are no laboratory methods that measure pesticide-specific exposures. And that's an important issue. At the meeting at EPA, there was a lot of push for EPA to require pesticide-specific biomarkers before a pesticide can be registered. And I think that's an interesting requirement, although when you look at some of the issues, which I'll raise in a second, there's some challenges there. The response to industry at that meeting was saying then EPA should start to allow human dosing studies again, because that's how they can really develop appropriate biomarkers and specific biomarkers. How to judge the expected validity of a biomarker if that does become a requirement. Right now, in our work and in, for example, the work out of Columbia, they're measuring chlorpyrifos at the part per trillion level in blood and seeing relationships with health outcomes. When these pesticides were registered many years ago, nobody would have considered that a part per trillion measurement in blood was even possible and that it would even be meaningful for a health outcome. So, and if these are ultimately found to be valid, I think we need to rethink risk assessment and how incredibly low levels for things that are out in common use may have adverse health impacts. So in terms of the future, I think we need more research, particularly on intra and inter-individual variability of exposure markers. I think there is a need for more work on pesticide-specific biomarkers, and we also need alternative dose metrics, and maybe Elaine will talk about some of this, but perhaps looking at other kinds of markers, omic markers, or things like cholinesterase-bound functional OP groups as a way to really quantify the exposure to the parent compound that can be distinguished. And I'm not going to talk about peroxidase or other factors here, but there's a lot of other intervening individual factors that can affect how you interpret those results. So I have a few minutes here, and I'm just going to highlight now, this is kind of more an advertisement for some of the other work we're doing and just highlight some of the issues, although I think some of the work we're doing on childcare might be relevant now to some of the issues that George raised. I want to mention here that we have this issue of exposure to how to interpret urinary biomarkers, we did some kind of pioneering work working with CDC to develop measures of non-persistent pesticides in breast milk. And we do find chlorpyrifos and other non-persistent pesticides in breast milk. So we know kids are getting exposed to it. And maybe breast milk could be a good direct marker of exposure to the pregnant woman and maybe to the very young child. Although, again, we know as the child has other sources of exposures, and there's always that challenge of trying to distinguish dietary and non-dietary exposures. But we are required to assess dietary exposures, and usually, at least EPA, does not think about breast milk for many of these non-persistent compounds. So this is another area we're working on. Another piece we're working on in terms of exposure biomarkers, I can give a whole talk on this, is looking at manganese exposure as a marker of fungicide use in California. A lot of fungicides in California, Maneb and Mancozeb, have a lot of manganese in them. In fact, the biggest source of environmental manganese in California is agriculture. And we're working on a project to measure at very small increments, like 20 micron increments, manganese in deciduous teeth in children. And interestingly, there's something called the neonatal line. When children are born, there's a change in the alignment of tooth crystals, and that creates a discontinuity that actually creates a line in the tooth. And if you measure manganese on different points of the line, you can actually distinguish exposure prenatal and postnatal. That's another kind of biomarker piece that we're working on. It's fascinating. Hopefully we'll have some papers out on it soon. Another piece of work we're doing is on childcare, and we have a whole project to improve environmental quality in childcare. We've been working with DPR and done some surveys in California on pest management and pesticide use. A lot of pesticides are used in California childcare facilities. About half are using them to control pests. About 20%, not on this slide, are using them monthly to control pests. In some cases where there's no actually identified pest, it's just being used prophylactically, essentially. We've also have some funding from CARB to do fairly intensive environmental monitoring, and I decided that the data is just not quite far along to do a whole talk on this, but we've been measuring a bunch of different both volatile chemicals and things like phthalates and flame retardants in air and dust. Just to highlight a few things we found. We've quantified over 70 volatile organic carbons in indoor environments. Similar to other residential environments, we find much higher levels indoors than outdoors. Here's an example of D-limonene, about 33,000 versus 800 nanograms per cubic meter. Levels so far have been in the range of homes and other buildings for many compounds, but childcare is unique in the high level of both use of cleaners and disinfectants, much higher even than regular school. So there's potential for relatively unique exposures that go on there. And then, of course, there's all the behavioral things about kids that make them more exposed to anything that's their environment. We also found that when we were looking at VOCs, there's, in some cases, up to 60% of the total volatile organic carbon mass was not measurable. So we went through our chromatograms and linked the unknown peaks to a NIST, National Institute of Science and Technology, spectral library to identify these unknown compounds. And we went through a process, I can't really go into the details here, but we've also identified 130 additional chemicals that are likely present. So in this small study of 40 facilities, we found over 200 chemicals in air, some of which are potential concern in terms of child health. We found lots of flame retardants in dust in these facilities, both brominated PBDEs, also chlorinated tris, and also Firemaster 550 breakdown products. These median levels tend to be lower than some of the residential levels reported elsewhere in California. The peak levels, though, are right in range, and there's not too much data out there for either the Firemaster 550, or if you'll notice here, 209, which there's not much data for either, was also widely detected. All of these had 100% detection in childcare facilities, and we looked at PBD 209. This olive green here is the relative proportion of PB 209 in the 40 samples we measured. So basically, for example, in this sample, about 80% of the total flame retardants in that sample were 209, and in this sample, only about 10%. So you can see in general, if we look at, this is 209, this one's 47, and this one's 99, these three flame retardants made up the greatest proportion of the total flame retardants in these samples, these dust samples. The penta mixture was about half, but the 209 is also about half, even a little bit more of the total flame retardant load in these dust samples. 209 is pretty much unstudied in California, and I think it's something we need to look at in more detail. Quick summary on the child care work. There's lots more to do, and there's a lot of, I think, rich and important research to look at in understanding the unique exposures in this environment. So that's the end of my talk. I want to thank our funders. The CHAMACOS work and center work is primarily funded by EPA and NIEHS, and we've also had support from NIOSH. And then the work we've been doing on child care has been supported by the California Department of Pesticide Regulation, and also this report that we'll be coming out with soon from the Air Resources Board. So there's kind of a smorgasbord of stuff at the end of this talk, but I hope putting it together, there's some interesting issues to talk about. Thanks.